So in last unit we talked about chromatin and chromosomes, and briefly the difference between them was that chromatin is how the form that DNA is normally in during the cell cycle, and chromosomes is how it is in a short period of mitosis, primarily because the chromatin coils up and chromosomes are more denser form. So we're going to kind of zoom in that a little bit deeper and introduce a new term here, and the goal for today is to really introduce a handful of key vocabulary, which we'll show right here, walk you through them, and that by the end to have a deeper sense of these terms. So let's get started. So let's look at the nucleus of a cell, and all this image in here that I've drawn, imagine, is chromatin. So that's kind of like that spaghetti noodle shape that we say that the DNA is normally in during the cell cycle. Now, if I asked you how many pieces of chromatin are there, it's very hard to tell in this shape. Is this, 2 really big pieces? Are there 7 smaller pieces? And each organism has a certain number of pieces of chromatin. So imagine that the way we're going to figure this out is I took them and I spread them out. I pulled them all apart and laid them out one by one. And if I did that, it looked like this. And now we can see exactly how many pieces there are, right? There are 4 individual pieces of chromatin, right? 1, 2, 3, 4. Don't worry about the color coding yet. We'll get to that. Now, a couple points we want to make about the DNA in the nucleus in general. And the first is that if you look at all these pieces, none of them are identical, right? These two have a different color, and these two have a different size. However, each piece of DNA in a nucleus has a pair. So these two are pairs, and these two are pairs. And when you look at the DNA, one of those parts of each pair came from mom, and the other half came from dad, which was why I'm kind of coloring it like this. So let's just kind of go with these stereotypical colors that represent biological sex and assume that this blue DNA you inherited from your father and the red DNA you inherited from your mother. All right. Now, these 2 pieces of DNA that are very similar, we call homologous pairs. Homologous meaning similar, and pairs because there's 2 of them. So in this picture, we have 2 homologous pairs. Now, what we want to know, and this is one of our key vocab terms for this talk, is what is a homologous pair? What defines it? And it's 2 ideas here. The first is that they have the same genes, and the second is those genes are in the same location. So let's walk through an example of that. So imagine on this piece of DNA, there's a gene for eye color, and it's located right here. So what must be on this part of this homologous pair? Well, since the definition is that they have the same genes in the same location, there must be a gene for eye color located in that same area. Right? Now, on this piece of DNA, let's assume that there was a gene for blood type here and another gene for hair type here. So what must be the genes located right here? And we see again, it's the same thing. We need to have the same genes in the same location. All right. However, what they don't need to have is the same alleles. All right. So as a quick reminder, alleles are variations of genes. So if there's a gene, so eye color is a gene. What are some variations of eye color? Brown, blue, hazel, green, etc. So those variations, those colors, brown, blue, green, are the alleles, and eye color is the gene. So when you have these different genes, they can have different alleles in those places. So for example, all right, let's say on this piece of DNA you inherited from your dad, you got the allele for brown eyes, but on the gene that you got from your mom, you inherited the color for green eyes. Down here we have the genes for blood type, but there are different alleles of blood type, so different variations, right? Like A positive or A blood type, B blood type, A, B, O. I'm sure you've heard of these. So let's imagine that from mom you inherited the A blood type, and from dad you inherited the O blood type. And for hair type, again, we have lots of different kinds of hair out there, right? You can have straight hair, kinky hair, curly hair, etc. And let's imagine that from mom you inherited curly hair, and from dad you also inherited curly hair. So keep in mind, the point here is that you could potentially inherit the same alleles from mom and dad, but it's not necessarily the case. You can also inherit different alleles. All right. So homologous pairs are not identical, right? And this is a key idea. They are just similar. And what makes them similar is that they have the same genes in the same location, but what makes them not identical is the fact that the alleles are not necessarily the same. The alleles can be different. All right. Again, that's a key idea that I will ask you again and again. I'll tell you, I'll ask a question like, okay, homologous pairs are similar, but they're not identical. Explain. And the key thing we want to say here is that what makes them similar is same genes in the same location. What makes them not identical are the alleles. All right. So now that we have covered some of those basic terms, what we want to do is we want to go back and review the cell cycle from our last chapter. So here I have the stages. We got G1, S, G2, etc. This is prophase, metaphase, anaphase, telophase, and cytokinesis. So we're going to walk through those again really quickly, but we're going to focus more on what's happening with this DNA in a little more detail than we did last unit. So in G1, the cell grows. Since I'm not really showing anything outside the nucleus, nothing's going to happen here. We're going to focus mainly on the DNA. So now in S phase, we see the DNA gets copied. So watch what's going to happen. Every single piece of DNA on the screen, I'm going to make an identical copy of. All right, do you see that? Let's watch one more time. So every piece of DNA gets a copy. Now, we go through G2, and we have the organelles copied, and then we get into the prophase, and the nucleus disappears, and then also, the chromosomes form. Right? So we said that chromatin coils up to chromosomes. And this is the part that we need to discuss in a little more detail. So bear with me here as we go into some of that nomenclature. Okay, so each of these are all strands of chromatin. And to make a chromosome, a strand of chromatin just needs to coil up on itself to become a more tightly wound up form. So it would look something like that. That top piece used to be a long strand of chromatin, and now it's curled up into this more denser packaged chromosome. All right. So if I asked you, what do we call this? This would be a chromosome. What do we call this? This is a piece of chromatin. Now, each strand of chromatin coils up like this. Now, if I stopped you right here and I said, wait, how many chromosomes are present? How many pieces of chromatin are present? The answer to this would be 4. There's 1, 2, 3, 4 chromosomes. And there are 4 pieces of chromatin. 1, 2, 3, 4. All right. So all of them coil up. And now again, if I asked you, how many chromosomes are now present in the cell? The answer would be 8. 1, 2, 3, 4, 5, 6, 7, 8. And how many pieces of chromatin? There are 0. Now, some key things to remember here. These two chromosomes are identical. That's why I've drawn them in the same color, in the same shape, same size. But these 2 chromosomes, this red and the blue one, these are homologous, right? So they represent that pair of chromosomes that are similar, same size, same genes, same locations, but their alleles may have been different. Okay. Now, I mentioned that because what happens next in prophase is the identical chromosomes come together, right? So what would that look like? It looks like this. And once those identical chromosomes come together, there's a special structure called a centromere that attaches to them and holds them together. We kind of think of the centromere as like a paper clip. All right, so it's going to keep these pairs of chromosomes together. Now this step is also where the naming gets really, really confusing. So to simplify this, I'm going to look at just one of these pairs. So let's get rid of all of these except for this one right here. All right. Now, when these were separated, right, we called them a chromosome, right? This was a chromosome and this was a chromosome. There were 2 chromosomes. However, when we fuse them together, what we now do is we call the whole structure a chromosome, and we call these 2 pieces chromatids. And oftentimes they're called sister chromatids. So again, that's this very confusing step, right? The word chromosome can mean multiple things. It can refer to them individually, like right here, this is a chromosome, this is a chromosome, or it can refer to this whole structure of 2 sister chromatids fused together. All right. So again, let's kind of review the difference between a chromatid and a chromosome. So imagine again I gave you this on a pop quiz or a warm-up, and I draw these structures and I say I want you to label these. What do we call this structure called here number 1, 2, 3, 4, 5. And so the answers would be, and go ahead and pause this video and try to take a guess first before I show them to you. But the answers would be this. Number 1 is a chromosome, right? This whole thing here is called a chromosome. And this individual strand is called a chromatid. And the piece that holds it together is a centromere. However, if they were separated, then we would call each of them their own chromosomes. That word chromatid we don't use unless it's this connected piece of these two identical chromosomes. All right. So if I asked you how many chromosomes are there on this screen, the answer would be 4. 1, 2, 3, 4. And if I asked you how many chromatids, the answer would be 8. There's 1, 2, 3, 4, 5, 6, 7, 8. All right. Now, that's really the confusing part of all of this. So the rest of the cycle will be relatively straightforward. So in metaphase, we know the chromosomes line up in the middle. In anaphase, they get pulled apart. I'm sorry, a pop quiz. So if I stopped you right here and I said, all right, on this screen, how many chromosomes are present and how many are chromatids? Go ahead and pause the video and take a guess at that before I tell you. And so the answer would be there are 8 chromosomes and there are 0 chromatids. Because remember, chromatids only exist when they're in this fuse stage. All right. So now we go into the next phase of telophase, and we know that chromatin forms, right? So those chromosomes uncoil each of them, and then the nucleus forms, and we're back to the end, and that would be the end of the cell cycle. Okay. So that goes through our talk today, and these are the key terms that we want to understand. So what is a homologous pair? What is an allele? Centromere, chromatin, chromatid, and chromosome. And you want to go back and watch this video again until you feel very comfortable understanding the definition of each of these terms, right? And be able to explain them in context.